We actually are getting knowledge that these particular, in any complex ecosystem, a lot of these things, if you like to use the terminology, talk to each other. They synchronize. There's also a lot of competition going on between them as well.
What many of us see as problems could also be big opportunities.
The big inefficient one that sticks out is the production of methane. While ruminants lose anything up to about 10 to 14% of their entire energy that they produce in methane going out to the atmosphere, other animals that have what is apparently a similar type of digestion, it's not the same because those animals did evolve entirely separately. But the thing is that the macropod kangaroos have a foregut fermentation. So it's the same sort of system. But the interesting thing was that it had been reported that kangaroos, particularly larger kangaroos, didn't produce methane. Is it possible to get a similar, or to be able to move the microbiome in sheep and cattle to a situation where they can do a similar thing?
Could nature provide the map to the elimination of livestock greenhouse gas emissions?
And we have to remember, it's not the only ecosystem the only fermentative ecosystem where reductive acetogenesis does dominate. Wood-eating termites, a reductive acetogenesis tends to dominate in their hindgut. It dominates in the hindgut of ostriches as well. It also actually dominates in about 2/3 of humans. Only about 1/3 of humans are methanogenic.
Well, good morning, Athol, and thank you very much for joining me today. Could we just start with the background of how you got into researching the microbiome of ruminants and other species?
Well, I guess the whole point about animal agriculture and trying to improve animal agriculture in the ruminant space is that those animals are entirely dependent on the biome, the microbiome in the gut, and in particular the rumen microbiome for virtually all of their energy and protein requirements. So it's a necessity to know and understand how the microbiome in those animals works. And indeed, as we're finding out more and more, how the microbiome in all animals works.
Where do you see us now in terms of how much we still have to know compared to what we do know?
Basically, we know a hell of a lot more. We know a lot more basic information, but we're also learning that we also need to know things that we hadn't realized before, like how organisms communicate with each other, how they cooperate with each other when they are, for example, using metabolites and the like. We actually are getting knowledge that these particular, in any complex ecosystem, a lot of these things, if you like to use the terminology, talk to each other. They synchronize. There's also a lot of competition going on between them as well, and we need to understand that, because if we're looking from an agricultural perspective as opposed to a natural ecosystem perspective, what we're trying to do is to direct the metabolism in such a way as to maximize the efficiency for what we want, which is the efficiency of, say, degradation of plant fiber, for example. It's not saying that the process is inefficient as it is, but for our particular targets or goals in this area, if we can manipulate those to reduce some of the what we would call inefficiencies, and promote the direction that we're going in. That's basically what we're trying to understand. So our ability to understand everything in such an incredibly complex system is probably still a fair way off. But to understand enough about aspects of the system to be able to improve it in the direction we want to improve is well within our reach.
That's very exciting to hear. I think in some of the other people I've spoken to, and they've alluded to what you said about the microbes communicating and actually collaborating with each other.
Balance.
What's more important, having the species in the rumen or present, or
no,
how they are actually interacting and collaborating?
That's a good question, because I think it's how they're interacting and collaborating. There's a lot of what would you call it? Inbuilt redundancies in the organisms that are present and what they do. So basically, if you, for example, you might have a dozen different strains of a certain species. If you knock out a number of those, others can take over that particular role in terms of the the specific breakdown of certain products and use of certain substrates that they do. But it's how they communicate with each other and the effectiveness with which they do it. You might have, for example, an organism that is dominant because it's able to outcompete other organisms, but it only breaks down the material quite slowly. So it breaks down cellulose quite slowly. Whereas if you could influence another particular species that breaks it down quickly but isn't as competitive, to become more competitive, then you would be getting an advantage coming through there.
In higher organisms, metabolism changes with age, it changes with levels of stress, it changes with temperature, humidity, etc. How are microbes affected by the age of the microbe or the levels of stress they're under? Does that change their metabolism, or is that something we even know?
It's both absolutely, and we don't necessarily know at all. It certainly is. Now, the age of organisms is an interesting point, given that organisms reproduce by binary fission. So they're splitting in half, and then you got two new cells. But it has been shown that some older cells, and some older cells will die, actually do change their metabolism. But they also change their metabolism with temperature, with metabolites present, with osmolarity, with just about everything that you can think of. But the really interesting one is also they will change their metabolism, some of them, with a rate of growth. And the rate of growth, as affected by, say, washout from the rumen or whatever it is, or the amount of substrate that's available to them, can affect markedly the type of products that they produce. They can actually even switch to different genetic pathways at different rates of growth. A really good example of that we have seen, and we did some work on this in Illinois back in 2014, was with Streptococcus bovis, which is a known lactic acid fermenter and or lactic acid producer, sorry, and it's always been a problem when it's growing really, really fast. So that some animals just gone onto a grain diet, it will produce just about pure lactic acid. It's a homolactic producer. But if you go back to an animal that's on a forage diet, the strep bovis populations are actually about the same. They don't really increase that much unless the animal goes acidotic, then they go over the roof. But the difference is that they produce very little lactic acid. They produce other metabolites, acetic acid, and I think a bit of propionic and some others as well. So you can change that. But the other big interesting thing that we're only just getting a handle on, and this is really the whole of animal biome, if you like, is the interaction between the animal itself, the physiology of the animal, which, as you rightfully said, will change with all sorts of different things. But that also interacts directly with the microbes. So directly through salivary inputs, through materials coming in through the rumen wall, etc., probably temperature changes, all sorts of other metabolite changes. And we don't really understand how that interaction occurs and what we can do about that. There's also the genetics of the animal. The genetics of the animal appear to have an impact on the type of microbes that are able to colonize the gut as well. And that would be another area that would be particularly of interest to understand how that occurs.
At some stage through your career, you started looking not just at ruminants, at cattle and sheep, but at other species. Do you want to just tell us a bit about why you
really,
explore
really, I mean, the major other species, we did look around at some other species just from the point of view of interest in what type of microbiome they had. But the major ones that we're really talking about here are kangaroos. And really that goes back to the whole methane issue. So basically, when we're looking at improving the efficiency of the microbiome and the rumen and metabolism, the big inefficient one that sticks out is the production of methane. Methane is energy rich. If instead of producing methane, we could channel the substrates that produce that into other usable substrates, and the obvious one here is a process called reductive acetogenesis, where acetates produced, which is a major VFA that's used for energy by the animal. Okay, so it came to our interest, and it was all from the foregut fermentation. So from the ruminant perspective, that while ruminants lose anything up to about 10 to 14% of their entire energy that they produce in methane going out to the atmosphere, other animals that have what is apparently a similar type of digestion, it's not the same because those animals did evolve entirely separately. But the thing is that the macropod kangaroos have a foregut fermentation that is functionally similar to the rumen. It's a huge chamber that microbial activity breaks down forage, breaks down plant material to produce VFAs, and the material flows through then into the true gut, where the microbes, etc., are digested, and that produce the protein for the animal. So it's the same sort of system. But the interesting thing was that it had been reported back probably in the late 60s, 70s, 1970s, that kangaroos, particularly larger kangaroos, didn't produce methane. And so that is really what fueled our interest in looking into other animals, is why that happened. And if that happens in a foregut fermenting animal that feeds on pasture, and often with the grazing kangaroos, the very same pastures that cattle and sheep are feeding on, then is it possible to get a similar, or to be able to move the microbiome in sheep and cattle to a situation where they can do a similar thing, and therefore that 10 to 14% energy, instead of going out in the atmosphere and causing or contributing to issues that we have with global warming and climate change, if we could channel that through into growth, improved growth, then we'd be getting a lot more meat or milk available from those animals without any extra feed being required. And that's where that was really our driving point to look at kangaroos.
So where's that at, and what learning did you find, or what would be the next steps there?
Yeah, well, we finished I finished at a point there where we were able to introduce stable isotope probing or stable isotope tracing, and we were able to show definitively that the precursors of methane, which is carbon dioxide and hydrogen, in cattle, the stable carbon goes into methane, as you would expect, and out that way. But in kangaroos, it ends up in acetate. And so we're able to actually show that the predominant method for removing hydrogen in particular from the fermentation in kangaroos is reductive acetogenesis. And we're able to then go through, and we're able to isolate I think 2 or 3 species of reductive acetogens that utilize carbon dioxide and hydrogen very efficiently indeed. and they weren't known species. They hadn't been cultured before. But we're also able to show that that was basically the tip of the iceberg, and that there were a whole range of other bacterial species in kangaroos that were basically reductively acetogenic. So they were forming acetate from hydrogen and carbon dioxide. So where we were thinking of looking at, well, there's two approaches. One is hopefully we might be able to isolate those microbes, and hopefully they might be able to colonize or persist in the ruminants. Now that is probably a long shot on its own, but it's probably worth having a go. The other thing is to try to understand what conditions or what set of circumstances or what other microbes are required to enable them to dominate in, say, the kangaroo foregut when they don't dominate in the rumen. They do occur in the rumen, but they don't dominate. Yeah, so that's where the directions, and the main direction we were interested in was to understand how that can happen in kangaroos, so that we might be able to emulate similar systems, similar circumstances in the rumen to generate that advantage to the reductive acetogens.
So that goes back to understanding how the interactions between those bacteria and the other bacteria that are present, plus any potential, I guess, communication molecules or signaling molecules or systems that turn them on or turn them off or tweak their metabolism.
Yeah. And it may be more complicated than that, because it might be that there are certain microbes that actually protect them in that situation as well. So, you might have ones that they work in synergy with, but also ones that are able to be antagonistic to, say, methanogens and that type of thing in the rumen as well. And we have to remember, it's not the only the only fermentative ecosystem where reductive acetogenesis does dominate. And there could be interesting issues about genetics in that as well. For example, wood-eating termites, not detritus-eating termites, but wood-eating termites, a reductive acetogenesis tends to dominate in their hindgut. It dominates in the hindgut of ostriches as well. It also actually dominates in about 2/3 of humans. Only about 1/3 of humans are methanogenic. So, what switches it on, what switches it off? Can we work that out? Can we look for those particular triggers, those particular switches?
You mentioned a number of species: termites, humans, ostriches. Are there any patterns across nature that sort of say that this type of animal, this type of ecosystem favors a certain type of either acetogenesis or methanogenesis?
It would be lovely if there was. And I reviewed a lot of this literature quite a while ago now and looked through it in a great deal of detail. And realistically, we were not able to come up with any definitive factors of size, invertebrate or vertebrate, whether they're warm-blooded or cold-blooded, even whether they're carnivorous or herbivorous. They suggested that. I mean, one of the big issues that always comes is rate of passage might have an effect. Well, it might, but in a lot of these animals you've looked at, it's really not a big issue. The temperature isn't an issue either. It's interesting that, for example, some of the very large snakes and crocodiles, which are totally carnivorous, actually produce methane. Whereas a lot of other, plant-eating insects, kangaroos, ostriches, etc., which are basically herbivorous, don't produce methane. And yet then we've got, a lot on the other sides of that as well that do produce methane that are totally herbivorous too.
You mentioned humans were roughly 2/3 acetogenic and 1/3 methanogenic. Are there other species where there's a divide like that we're aware of?
Not that we're aware. I don't think there are that we are aware of, which is sort of interesting in itself. Now, I think I couldn't put my hand on the literature, but I think I have seen that there could be genetic reasonings between this in humans as well. And basically switching diets, for example, won't change whether they're methanogenic or reductively acetogenic, which is quite interesting in itself.
I guess there's two things that come from that. The first is age and the time that this is set, and whether it's something that's set very early in life or even in utero, and that has an effect. And the second thing is if there's not really any clear patterns within nature. Some animals are acetogenic, some are methanogenic, and then there are species like us that can be either or. Is it, I guess, naive to say that it's not fixed in nature, so it can be changed, and the different species are a map that nature has provided, and our challenge is just to work out how to read that map?
That would be one way of looking at it, and that would be a definite possibility. I mean, it's really difficult to say why some species of animals are methanogenic and some are reductively acetogenic. Also, whether it can be changed is an interesting issue, and when it can be changed. Now, on that area there, we had done some research as well to look at whether we would be able, from a general perspective anyway, if we're going to look at trying to manipulate the rumen ecosystem by introducing microbes and that, we know that if we have an empty or a blank niche in there, that we can put something in, such as Synergistis jonesii when we introduce animals to leukemia. Or if they change diets, and we can introduce something that would already come into the ecosystem. That's just an example that if there's nothing else in the niche, then you can put something into that complex ecosystem and it will do it. But introducing organisms into a complex ecosystem and trying to get them to compete with organisms that are already, with it undertaking a role there, and you want to try to move them out and move these ones in, could be considerably more difficult. And trying to work out, okay, no matter what you're trying to manipulate in the rumen, when is the best time to try to get a permanent change made? It's pretty obvious, when animals are born, their gut is sterile, or effectively sterile. And then it develops, and it develops quite quickly. And it's just when, which organisms get in there. Are some intermediates that, are taking it from one particular level of nutrition to another level of nutrition. So when can we actually introduce those particular microbes in there and get them to stay there for the whole thing? That it's a big question, and it's an important question if we're ever going to try to successfully manipulate the rumen by, for example, I'll go back to fiber degradation again. For example, if we want to introduce a more highly fibrolytic microbe into that, do we need to get that in before anything else is in there? Can we knock something else out by getting it in later on? That's quite an issue.
So where do you see the next steps in terms of understanding how, I guess the next steps, not just in understanding, but in making a tangible step in terms of the rumen microbiome fermentation towards something that's more acetogenic and less methanogenic?
Yeah. Well, I guess it does come down to that understanding, and there's two directions where we could go with that. One is the trying to introduce the microbes, trying to introduce from other ecosystems where we know, that they are dominant, and get them out of those ecosystems and get them into the animals at at an appropriate time in the life of those animals and see if that can work. But the other thing is actually understanding whether just simply changing some of the, or trying to change some of the some of the conditions that are occurring within the ruminant itself might enhance that. So more of a, so it's really a probiotic, I guess, and a prebiotic type of approach, where you're going to be not just having the correct microbes present, but also having the correct conditions that are going to allow them to proliferate and maintain those populations for a long period of time.
Excellent. Is there anything? Thank you very much. Thank you very much, Athol. Is there anything else that we haven't covered that you think is worthwhile mentioning?
But yeah, I think the thing also is that if we're going to get the value in terms of nutrition and feeding animals from reducing methane, we actually have to channel those products into something else that the animal can use that has an energy value to it. So, you know, the whole idea of just blocking methanogenesis won't really give you any benefit in terms of production.
So that then becomes critical in terms of what's the value for the producer if they've got an extra cost. And yes, there's an environmental climate impact or benefit, but there's no direct benefit for them. Plus, you've got the fact that there's, I guess, if it's a 10 to 14% energy cost, it's a bit like having a leaking tap. Yeah.
Well, you know, I remember saying to producers, you know, about, you know, why you should be looking at channeling methane into something else. And, you know, you grow all that pasture out there, you grow all those crops out there to feed to your animals. If I just went out and said, okay, take you know, a tenth of that out and just burn it and throw it away, you wouldn't be real happy about it. But that's exactly what you're doing at the moment. You know, basically, it's, you know, that is just going. And so if you want to get the value out of that extra material that you're growing to feed to those animals, you know, you want to make sure it's not going up into the atmosphere.
Well, thank you very, very much, Athol. It's been an absolute pleasure speaking to you.